This video is made for molecular diagnostic class explaining the polymerase chain reaction and COVID-19. COVID-19, first identified in December 2019 in Wuhan, China, quickly became a global health crisis. Initially linked to a seafood market in Wuhan, where early cases hinted at animal-to-human transmission, the virus soon spread worldwide, causing clusters of pneumonia cases, initially of unknown origin, later identified as COVID-19. This virus highlighted the crucial role of molecular diagnostics in public health responses. To diagnose COVID-19, usually a rapid test or PCR is performed. The rapid test is the one we can buy over the counter and use it at home. They come with instructions for self-collection of samples, usually via nasal swab or saliva. The results are generally available within 15 to 30 minutes, depending on the type of the test. The rapid test is an antigen test, checks for proteins that are found on the outside of a virus. The test strip acts like an antibody. If there are any COVID-19 antigen in your body, the molecule in the liquid will attach to those antigens and the line will appear on the test strip. This means you are positive for COVID-19. If there are no antigen detected in your body, the liquid does not respond and the line will not appear, often meaning you are negative for the virus. Benefit of rapid test is fast and easy that you can do it at home. However, the antigen test can only determine if you have an active virus in your body. The rapid test can't detect small amount of the virus. The rapid test is less accurate and there is a greater chance for a false negative. PCR test stands for polymerase chain reaction and usually performed at the hospital or in laboratory. PCR is a widely used technique in molecular biology for amplifying and detecting specific sequence of DNA. However, COVID-19 and some viruses does not have DNA. They use RNA as their genomic materials. Therefore, when applied to COVID-19 testing, PCR is used to detect the presence of RNA, the virus's genetic material. This technique is called the reverse transcription PCR. PCR is a molecular technique that replicates specific DNA sequences. It requires several components: a DNA template containing the target sequence, short DNA primers that attaches the target region and define a segment to be amplified, a terminally stable DNA polymerase enzyme responsible for synthesizing new DNA strands, deoxynucleotide triphosphate as building blocks for DNA synthesis, and buffer solution to provide the optimal chemical environment for the reaction. The PCR process involves several temperature control steps: denaturation, annealing, and extension. Denaturation: the reaction mixture is heated to separate the double-strand DNA into single strands, preparing them for subsequent processes. In the annealing step, the mixture is cooled to allow short DNA primers to bind specifically to complementary sequences on the DNA template. These primers serve as a starting point for DNA replication. Once primers are bound, the extension or elongation steps begins, where DNA polymerase adds nucleotides to primers, extending them along the template DNA. Each cycle of denaturation, annealing, and extension exponentially amplifies the target sequence of viral cDNA. To measure the accuracy of tests, sensitivity and specificity are calculated. Sensitivity refers to how well a test can identify individuals who have the disease, which is true positives. RT-PCR have proven to be highly sensitive in detecting COVID-19 cases, including those without symptoms who might unknowingly spread the viruses. Validation studies by Corman have shown sensitivity rates ranging from 85 to 100% depending on the amount of the virus present in the sample. In contrast to sensitivity, Specificity refers to the test's ability to accurately identify individuals without the disease, which is the true negatives. This specificity ensures that individuals do not have COVID-19 are correctly identified as negative. Systematic reviews consistently report high specificity estimates exceeding 98% for RT-PCR assays used in COVID-19 diagnosis. In conclusion, molecular diagnostics, especially techniques like reverse transcription PCR, have emerged as indispensable tools in the global response to COVID-19. By enabling rapid and accurate detection of COVID-19, these methods have significantly contributed to early diagnosis, prompt isolation of infected individuals, and effective implementation of public health measures.